To kick off the second half of our presentations, I would like to welcome Dr. Kevin Vilaca. Kevin is a veterinarian and a partner at Southwest, and he holds a master's degree in epidemiology. Today, Kevin will be talking with us about autogenous vaccines. Autogenous vaccines have come a long way, even in the time that I've been part of the veterinary world. One of my experiences as a vet student was at a cattle practice in Ireland. One of the strategies there was to take a papilloma, which was a growth caused by a virus, cut it off of a cow, chop it up, and inject it into the other animals, with the hopes that those animals will then develop an immune response and the impact of disease will be decreased on the farm. This strategy did work sometimes, but it did come with quite a bit of risk. I'm excited for Kevin to be able to explain to you and to give you insight into how we are helping farms develop autogenous vaccines and the process that is taken to make sure that they're not only safe, but also effective. Please help me welcome up Kevin.
You guys hear me in the back? Perfect. I like to move around a lot. So I get the privilege at this point to have a discussion with you about autogenous vaccines. And some of you have probably either experienced or seen it in the past or had it on your farm. I'm going to go through some of the idiosyncrasies, some of the things, and more importantly, where are we today? So I'm going to set the stage for you. And we'll start off with saying, so for most of you on your farms, you will have various diseases that you'd be dealing with. So we look at the big ones. Most of these meetings, most of the things that we would go to, you're going to end up seeing, well, we'll talk about PRRS, we'll talk about flu and myco and circo. And these are important diseases. These are things we spend a lot of time dealing with. And these are really important. However, when you deal with these diseases, what are the other things that you go and you have to end up actually treating with? And you look at these, not only are the prevalence of these viruses and bacteria very high, but they're not our only diseases that we deal with. So then we look at what about what we call our secondaries. So these would be bugs that would be normal inhabitants of the lung, yet they don't cause disease. So primarily for our worlds, strep and glassers are what we would end up seeing. When you're treating, you've got a PRRS break, you've got something going on and you're treating. Are you treating for the PRRS specifically, or is it going to be that pneumonia that's coming in, that strep, that heavy breathing, and you're going to put antibiotics in? Those are targeted for your secondaries. So you take the cute little pig over here in a bubble. What is that? Old study a long time ago when PRRS first showed up, where they took high health cesarean-derived pigs, put them in a bubble, and gave them PRRS. These pigs, yep, they got sick, but they didn't die. Now, they took those exact same cesarean-derived pigs, put them in a bubble, gave them PRRS, now gave them strep and glassers, and now they start dying at really high rates. And these are what we deal with on a regular basis. And so a lot of what you end up putting your time into is actually targeted at treating those secondaries. Although the PRRS break that just happened, the flu that just came in, are still your primary diseases, but it's those secondaries that we end up dealing with. So the shift really where we've gotten to is saying we've got complex systems. We've got diseases out there that aren't just one disease. For most of it, if we call it conventional Ontario health, we're going to have multiple diseases. Everyone's going to have some strep or glassers that they own in their herd. And so now where we go to is, yes, still deal with those big diseases. We've got great programs that you have to put in place to try to control those. But now we're looking at it and saying, what about a better understanding of those smaller bugs? Which ones are they? What are they doing? Where are we affecting? And even within the different strains, what are we seeing at different stages of production? Add into that sensitivities that then, yes, antibiotics that are used, we can target and actually put in place. But now we know which is the better antibiotics. We're putting a lot more strategy to that. But now the shift has come, which is looking at something like autogenous and saying, well, what about using the immune system? What about actually making it so that the animals that we're dealing with have the best coverage? They're still going to see PRRS. They're still going to see some of these other bugs that we have. But how can they handle it better? And can we shift? Can we actually shift away from some of the antibiotics that we're using and start using the immune system to get the best coverage that we can? I'll get Larry to start the short video. It's about 8 minutes. Please enjoy. So autogenous vaccines, what are they? Well, really, more specifically, an autogenous vaccine is a vaccine that is custom made for a herd from the bacteria that are isolated from that specific herd. And what that means is you get a perfect match to the bacteria that are in your herd causing issues. Now, autogenous vaccines are not new. They've been around for a long time, and some of the use historically was a bit of hit and miss. Fast forward to today, and that has changed significantly with the world we're in. We have diagnostic tests that we did not have before, which tell us so much more and have such better sensitivity. We've added in that the adjuvants, the parts of the vaccine that actually stimulate the immune system, now have much better technologies. They can actually target specific bacteria to create a better immune response to. Add into it with some of the computer sequencing, where we can actually go in and identify the virulence factors. This now gives us much better pictures on what animals have, what we're using, and then what to include in the vaccine. So we're not just throwing a lot of different bugs that we grew, we're actually putting the right ones in. We've also now moved on to the point where even our sampling that we're doing, the lab testing, is much more methodical and specific. The animals that we select for the testing are critical to making sure we get the right isolates to then put into the vaccine. And this as a whole has led to a global resurgence on autogenous vaccines as we've actually learned more about them. Add into that antibiotic use has been put a lot more pressure, and we want to reduce antibiotics. Autogenous vaccines have gained a much better positioning for that and have, with all these additions, given us much better products and with better efficacy in these pigs. How is it all performed and how does it get done? Well, come along with me and we'll show you how it's done. Sample selection is critical to getting a good autogenous. It all starts with making sure you've got the right animal that has this clinical signs, not one that is found dead, one that is freshly euthanized, as well as one that has not been treated. Since this is a sample, we will be going for strep suis. The whole heart and lung is removed while the pericardium is intact.
So I'm just opening a hole in the pericardial sac and taking a nice, clean, sterile sample around the pericardium and into the pericardial fluid, as Kathleen mentioned.
The pericardium is selected because of the fact that it should be a sterile environment. It should not have any bacteria in it. And therefore, anything that we find in it, if we've done good sampling technique, should be of significance.
I'm just plating it now on 2 different selective medias. And the pericardial sample is done.
Following that, we just take a closer look to see are there any other areas of interest or significance, and move on to the other part, which we have concern, which is actually a brain swab. Since this is a strep suis case that we will be going for, that is an important part. Right on the top, we do have the opening, and we will just go right into the spinal cord that can then be swabbed. So sampling is done across the top of the meninges, all through the areas in there to get a good representative sample of what's going on in there.
And again, the brain should be a sterile sample. There should not be bacteria in the brain in a healthy animal. So the presence of a pathogen such as strep would be significant and likely systemic.
So after the postmortem, we're going to work through looking for the actual microorganisms of interest. We're going to be targeting special organisms that we know are good for an autogenous, whether that's because they have many serotypes or they're specifically not a commercial vaccine available. And The next step is streaking the plates for isolation that were taken from our postmortem. The nice thing about doing this internally is we can be quick for our clients and we can be very targeted in the organisms we look for. We do that with both our blood and our chocolate plate. Our chocolate plate helps us find difficult to grow organisms like Glasserella parasuis, and our blood plate helps us see hemolysis. So an example of that would be like this. This is an Asuis plate, and you can see where you can actually see through the blood around the colonies. That's called hemolysis, and it helps us figure out which organism is important. So after we streak our plates, we'll take them to the incubator. We incubate at the same temperature as the temperature of a pig's body. And after 24 hours and 48 hours, we'll look for isolates that are significant and select them and transfer them to media yet again.
So once our microbiologists have pulled out the pathogens or suspect pathogens from the samples in our microbiology lab, they forward them on to our molecular lab for PCR testing. PCR testing is a rapid and quick answer to confirm the identity of the pathogens that we're looking for in swine. And PCR is really just a test to amplify a specific piece of DNA for that organism so we can identify that we have that specific pathogen. It's quicker and faster, and our veterinarians and our producers get answers in a very quick way.
So once the isolates are confirmed by PCR, we put them in the freezer for long-term storage. And that's done by making a lawn of bacteria and placing it in the cryo vial with the cryoprotectant for the freezer. So I'll use the swab to pick up a good quantity of the cells. And resuspend it in the liquid. And now they're ready for the freezer.
So if after watching this, you feel that an autogenous vaccine may be something that could be applicable to your herd, please reach out to your herd veterinarian and they'll be able to supply you with a lot more information and be able to advise you on how it might apply to your herd. So that was a bit of a tour of our lab, and mostly because we couldn't bring you all through the lab to take a look, but just so you have an idea of what the processes are that go in behind actually when those samples, we show up on your farm, we take samples, and then we give you a call back and you get some information. Well, there's a lot more that goes on behind it. And as you can see, there's a team of various people that are involved in getting some of those. So about 2 years ago, we started actually with this lab and started really ramping up. We've always had a lab, but started adding on more and more. And the biggest part to this is the goal of this lab wasn't strictly to be diagnostic. It was to be very targeted. And this is for autogenous autogenous bacterial isolation so that we could get the best samples out that then we could store and send to the specific labs that we then would actually have make the vaccine. And we saw the need for this mostly because we didn't want to just do everything. We didn't want to be able to, we've got some great labs, University of Guelph is phenomenal, but we wanted to be very specific. This is for autogenous production because it allows us to be very selective. So we've got over 255 cases since this has started. We've stored over 600 isolates, and no huge surprise, strep suis is 50 some odd %. That's still the majority of what's there, followed up by Glassers. And then yes, A. suis does show up, greasy pig, which is Staph hiacus, and then APP is still around. So these are probably the main ones that we would look at, with the top two being mostly where we targeted. So what do we see? For any of these, so again, specifically Glassers, that's GPS, and then Strep suis. There's a lot of serotypes. There's a lot of different ones out there, but usually some are more significant than others. And then even within it, these are what we're seeing as far as prevalence. So even though there are more than these, for us locally, we've got a handful of each of these that actually end up showing up. And some, and these ones that we've got in here, tend to be more significant and have some virulence factors or are known to cause disease. But we've got some variation within it, but not a huge amount of variation for what we see on a regular basis. Okay, so you think you've got a strep or a Glassers issue. Now what? Well, get a hold of your veterinarian and you say, hey, let's look into this. What's our next steps? The approach to this, I think if we go back and say, where were we in the past? A lot of it was we're isolating bacteria that come up, and if we find it, we thought it was significant. And maybe we're diverting it out a bit on which organs we found it in, but a lot was added in. And now we're getting to the stage where technology's advanced, we've had more experience with it, and we start saying, no, let's start looking at this in a more methodical way and use what we have available to us. So I say our Southwest approach because this is how we, and you've seen some of it in the video, how we would go about it. We believe that in order to get this, it's not one person, it's not your veterinarian showing up and collecting the samples and making the decisions. So it is a team approach. And so we will have individuals, we'll combine our molecular biologists, we'll go in and actually look at what we have for sequencing and actually then come together as a team to give you the answers that you need. We have rapid turnaround. So some of these, you can actually, today was one of the cases, went in, samples were collected yesterday. By today, I already have an idea of what it looks like is growing. Well, that's really nice as far as a very fast turnaround on what we think is actually going on farm. And then our ultimate goal to this is have it so that at the end of it, we actually have the right isolates that actually get put into the vaccine that are covering for what you're seeing. How is this done? Well, just like everything, there's pieces of the puzzle. And taking some of these individual pieces and adding them together, you may end up getting to where you need to be. However, it starts with what happens on farm. So we've alluded to it. What are the samples that we're seeing? What are you as a producer seeing on your farm, and where are most of the issues? Not an isolated case here or there, but what's predictable? What is actually happening on a regular basis? Then bring it back and do some of those specialized testing. Have the right growth media so that we know what is actually going to grow and target for the ones that we're looking for. And then multiple sample points. If you're going to do an autogenous vaccine, one of the worst things you can do is do it as a single time sampling. We actually want repeatability. And so for some of these, we're going until we get either a broader spectrum of what's there or we're getting repeatability. We're coming up with the same strains that are coming in. And that is very important. Take that in where we take some of the technologies. Full genome sequencing, where you actually look at some of these bugs and they now have virulence factors. So we can say not all strep suis are created the same. Which ones are they? And within those, do they possess the virulence factors? And those are done through sequencing. And then take this all together and say, with all that information, bring the team together and we sit down and we go through and say, based on what's here and how it grew and what it did on farm, what is the right fit and what's the combination? Because usually we're putting multiple isolates into a vaccine. So Glassers, a lot of times what we're going for is the heart. This is something heart, joint, pericardium. And again, it likes to grow on a chocolate plate. It's not actually chocolate, but we call it the chocolate plate. And once you get the little colonies, they're isolated out, checked. And then we get into PCRs to confirm, get isolates, and get our sequencing attached to that for virulence factors. Strep suis, slightly different. We like to get from the brain, pericardium. Again, these are areas where these bugs should not be. And I'll emphasize that because you can have a strep or a Glassers in your lungs or in the pig's lung, and it may not cause disease. So we actually want to see, is it in tissues where it shouldn't be? Shouldn't have anything in the brain, shouldn't have anything in the pericardium, but you might be surprised that you can find them, normal inhabitants of the lung, and not causing disease. Same thing, that's a blood agar plate, and confirmation by PCR, and again, further sequencing. Very targeted to the bugs that we're going after. So once we know what's there, there's commercial vaccines out there. And you might not look at it, so Serovar 5 and then the Suvaxin 1 is 4 and 5. These are actually account for about 50% of what we isolate, of what we've isolated in the lab. So if it's appropriate and it fits for your farm, go to a commercial vaccine if that's the strains that you have. Everything's great. However, it's usually not that simple and they're more complicated. And so if you've got any of the other Serovars, well, you don't have that as an option. You probably would have to look at going to an autogenous. Strep vaccines. Oh, there have been, when I first graduated, there's been 1 or 2 there were commercial vaccines. Challenge is there's a lot of variability within streps. There's, cross protection between it is limited, making sure you've got the right ones. Strep becomes more of a challenge. Add into it, in the past, we didn't have adjuvants that were very good at targeting and stimulating an immune response to strep, which we do now. There's been some great advances there. And so unfortunately, there's not anything commercial, and the reason for it is just too much variability. So why autogenous? Well, ultimately, the cocktail that you will have of different strains will be unique to your farm and may actually be specific to what virulence factors or what challenges you have. And so that combination that gets put in will be customized to your herd. The advantage to that is it's yours. It's not a similar one or some kind of like yours. It's identical. It was isolated from your pigs, from your farm. It's identical to what you have. So performance data. We ended up picking 2 herds out anonymously that we have within our database, and I'll give you some background to them, and they exemplify slightly different things as to what we expect out of it and what the procedure was. So case number 1, so this was taken from 7 different sites. So we did this over an extended period of time where we went to the various sites, isolated what was present there, and did a comparison across the system. What was repeatable? What were we coming up with? From there, we ended up going and saying, okay, what are the isolates that we've got? What's repeatable in it? And we've got some glassers, we've got some strep, and we've also got an A-suis. In this specific system, we're getting a lot of late-term losses, close to finisher. You've put all your money into these guys and they would get hit late. And it was actually a significant impact, but strep and glassers in the nursery was important. Again, that's sampling technique and making sure we're getting a good distribution of the system. So this vaccine was, because we were going after late finisher issues as well as some of the nursery ones, we ended up putting this in as a sow vaccination as well as a piglet vaccination. We don't do a lot of piglet vaccinations, but in higher challenges where, particularly in finisher, we need that, we may need to do piglet vaccinations as well. So nursery performance, and this one you look at, and in the pre-vaccination period, we had a lot of variability. We put our first batch in, and wow, it was great, it was working really well, and then we got hit with PRRS. And quite frustrating, and you see that I've highlighted that period in there where pigs were coming in at a much higher rate for what we were seeing as far as performance and mortality. And then once we got through that, it stabilized out to actually where we're currently sitting, where we're sitting at a much lower level, more like what we're seeing on the back end. What I can tell you is that even through this stage, what we ended up seeing, even with PRRS breaks, was much better than what we'd seen with previous PRRS breaks. We had some of that immunity on board. The other one I'll allude to here, and I'll get to it in more details, is antibiotic use. We were down just from that pre-vaccination to post-vaccination, the same time frame. We were down 66% in antibiotic use. And that's with the PRRS break in there where we did use antibiotics. Majority of our antibiotic use is in that shaded area. If you look at a monthly sales, that's where it was. Saying we never use them again, they're still targets. We were able to drop that by 66%. And then for our finisher, this is where we're seeing it. Our late finisher losses really dropped off. Those sudden deaths, those late treatments that we'd have to go in, really dropped off. So all around, we are seeing benefits. Now, we don't have average daily gain, feed conversion up here, but all of those have been impacted. The leveling out that we will see with the performance, instead of getting these ups and downs, it's a lot more leveled out on what we end up seeing on performance. Case number 2. This one exemplifies a slightly different one. So again, 3 cases were done. There was a lot more predictability within this one, and some glassers and strep was what was identified. Autogenous vaccine was put in place, and I'll show you the data in a bit, implemented, and this was just as a sow vaccination, so just given to sows on a pre-farrow basis. And what you'll sometimes see, because you've got a line there with 2023, you've got a line in there with 2024, what that shows is that what we will often see is that in our first round, we identify the common ones. You put a vaccine in place, you suppress those guys, someone else rises to the top. And in the process, they found one more that they've added in, and they've added in a suis. So this one's actually the next time they're ordering vaccine. I usually tell people try to order in six months intervals so that if something comes up, you can add it in. So their next vaccine will have this added in. But that comes back to some repeatability, some more testing, monitoring that goes on, and actually evolving. If you look at most vaccines that would be commercially available, they take years to develop. We're looking at months of trying to find it. It would make sense that we don't always get it perfect on the first try. However, by doing those repeated testing, we're much more accurate with that. And here we have it. So we've got the pre-vaccination. They didn't have the data in this prior. It was also very jagged. And once they put it in place, now this one, the mortality as a whole was lower, it's a lot more leveled out. It's a lot more stable. This specific herd, when we look at antibiotic use, they dropped their antibiotic use by about 50%. I think it was 48% that it dropped. And again, their level of use of antibiotics and what the growers are actually saying is it takes a lot less work. So I took these three studies, and the third one, they dropped by 30%. That was a simple one with just one isolate that was included. And then I threw in this fourth one last minute. This actually came back from one of our vets that said, yeah, we implemented this autogenous vaccine in place and was out at the farm, and they said in the last 3 months, they haven't had to use penicillin at all. And prior to that, they would have been about $700 a month just on penicillin control. So that's a significant drop, and it's a trend that we're seeing across the board. We're not saying you're never going to use antibiotics, but the reduction is it. So what have we shifted? If you come back to my start, the shift that we've moved towards is we're actually saying, let's use some of the other tools in the toolbox, not just use antibiotics to control these secondaries. Well, you'd have a PRRS break and you'd use a lot of amoxicillin to try to control it. What about other options? What else do we have at our disposal? So take-home messages. Today's herds are complex. They probably always have been. We keep adding in more disease. I remember from when I started practicing 20 years ago and what we have present, we seem to come up with, over the years, a lot more things that have come in, and we're challenged with more. And we've got the advantage technology has helped us in some ways where we've got more options to us. But it is more complex. We're dealing with multi-diseases at the same time and something that fluctuates. Having a good understanding of those little bugs. It's not just for an autogenous vaccine. It may be also for having a good understanding of what is my bugs, when are they hitting me, and what antibiotics are they sensitive to. Again, these are still tools that we have. Why not just add more tools to the toolbox? We've put a lot into a well-designed autogenous vaccine because that's the one we want is predictability. We want to make sure that what we're putting into it gives us the best return. And so that whole process of going through from beginning to end, all of those steps are critical. Just like those pieces of the puzzle, all those steps come together to give you the picture of exactly what the puzzle is, and that's what we can move forward with. So if this is something that you're of interest in, there's a lot more detail that goes into this and a lot more that may be specific to your herd. Reach out to your vet, ask him for any advice, any things, would this be applicable? It might be. It's not for everyone, but it may be a tool that you can have at your disposal. With that, I've got one more. I'm the one standing up here, but there's a whole group of people, whether you saw it through the video or some of the folks standing here. I'm fortunate enough that there's a bunch of people that have done some of the work in the background, the ones that actually collect this data, plate the samples, come in on weekends to replate the samples. Those are all things that they do. So for them, whether it's the lab folks, our research folks collecting some of that data, there's a lot of people that go into what I'm just presenting here. Rest of our Southwest vets, even that last little piece that came in on just this was really cool. I thought you might be able to include it. And that was just a neat little anecdote. And then our producers, phones are using it. And you guys for coming. You've taken your afternoon. Hopefully the information we've given you has been useful. And for that, I'd like to thank you. And with that, I'll take any questions.
So
you were going through, you're starting putting more, you put in some strips and some blasters in the same vaccine, right? I got that right. At what point could you go to a herd and find all the things we're vaccinating sows for specific on that farm and put it in one bottle for one shot for the sow for everything?
Great question. There is a limit. So you can go south of the border, and Marty tells a joke that he was speaking to a practitioner, and he got the 49th strain of something that he was putting in. That's probably to an extreme. There is a limit of what the immune system can see. And so you put too many strains in, and the magic number seems to be somewhere around 9. If you start getting too much more than that, you may not get the coverage that's with it. So is there an upper limit? You bet. Most of these, when we're searching for them, we're targeting specific ones. And so we're looking at it. And so very rarely would we have to get into those upper ranges. So yes, they could, but it's very rare. Most of our autogenous vaccines have 4 to 5 strains. A couple of streps, a couple of glassers. If you've got a suis or something else, maybe that's included as well. And again, you look at that prevalence of what we're seeing, we don't pull out a lot. 4 or 5 streps, 4 or 5 glassers make up the majority of what we see. That would be very similar to what you'd see on farm. Does that make sense?
Yeah, I mean, I'm just thinking because if we're using circle PCV2, PCV3, straps of blaster, scour vaccine, or syphilis, hurdle, like there's 7. You get all that in one sow shot.
Yeah, probably not. So reason I say that is you start getting it. Yeah, no, thank you. Because what you get into is different adjuvants, how we stimulate the immune system. All these different things start playing in. The reason I hesitate is you go south of the border and they'll do some weird things like that. Doesn't mean it's right, I guess would be it. So I would say you can still get coverage off of various diseases, but now you're doing your vaccination timings and splitting out your timing. So all in one, yeah, you're asking for trouble. But you could find someone that'll do it. And sometimes you don't have to go too far.
No, I think that's everyone. Thank you
All right,
again, Kevin.
thank you very much.